Early life exposure to the SSRI paroxetine exacerbates depression-like behavior in anxiety/depression-prone rats. Depression is one of the most widespread mental illnesses in the United States. The neural circuits involved with depression are thought to be sensitive to early life environmental factors, for example, exposure to stress and drugs, or maternal style and maternal mood. Women are also 70% more likely to develop depression than men, especially while pregnant and just after pregnancy. Most common treatment for pregnant women with depression is called a selective serotonin reuptake inhibitor, otherwise known as an SSRI. This is a type of antidepressant that works by elevating synaptic serotonin levels by blocking serotonin's reuptake transporter. Since 10 to 20% of women develop depression during pregnancy or directly following childbirth, thousands of children are exposed to SSRIs during very important developmental phases. This exposure may interfere with brain development. We can clearly see the SSRI blocking the active zone of the presynaptic nerve. This does not allow reuptake of serotonin and causes the serotonin to build up in the synapse. Previous studies show countless neonatal serotonin system abnormalities as a result of SSRI exposure. Some of these abnormalities include reduced serotonergic neuron firing, diminishing of the reuptake transporters of serotonin, reduced serotonin fiber density in the hippocampus, and altered monoamine levels in multiple brain regions. This study was broken down into 3 experiments. The objectives of the experiments are as follows. Experiment 1. Determine if treating adult rats that are prone to depression with SSRI paroxetine would improve depression-like behavior. Experiment 2. Determine if early life exposure to paroxetine would cause issues with depression later in adult life. And determine if rats that were prone to depression-like behavior would be affected worse by exposure than healthy rats. Experiment 3 will use genome-wide expression profiling to find molecular differences in the exposed rats. Important to note that in this study, BLR rats are rats that are genetically prone to developing depression/anxiety issues in adult life, and BHR rats are normal, healthy rats. The treatment, which was the SSRI paroxetine for all rats, was administered in the rats' drinking water. Experiment 1, adult male rats were treated with SSRI paroxetine and then performed multiple behavior tests to determine if depression-like symptoms were improved at all. Experiment 2, the rats were mated and pregnant female rats were treated with SSRI paroxetine while mating, during pregnancy, and during the postpartum phase. Once the male offspring of these treated female rats reached adult age, they were given multiple behavior tests to determine if BLR rats were affected more by the exposure than the BHR rats. Experiment 3, the exposed BLR offspring were decapitated. Different rats were decapitated at different ages to examine how the effects of exposure changed over time. Some rats were decapitated at day 7, some day 14, some day 21, and some were let to live to the adult age of 75 days and then decapitated. The brain tissue layers were harvested and stained, and then the stained tissue was analyzed using genome-wide expression profiling to find molecular differences. The graph shown here shows the results of a different behavior test for BHR and BLR rats given a vehicle, which is like the control group, and given the paroxetine. Overall, the BLR rats displayed more depression/anxiety-like behaviors than BHR rats. BLR rats given paroxetine spent less time immobile than the BLR control group. Paroxetine did not affect the distance traveled by BLR rats and barely affected the percent time spent in open arms. BLR rats given paroxetine had a slightly higher fecal production than BLR control group. This difference was insignificant. Experiment 2 shows that perinatal exposure to paroxetine caused an extreme increase in immobility in BLR rats. And remember, BLR rats are the rats that are predisposed to depressive-like behaviors. Overall, the BHR rats, or the healthy rats, were not affected by perinatal exposure. In these figures, the red points show changes in gene expression. As you can tell, early on, the amygdala was only minorly affected, but showed many changes in gene expression from day 14 to adult life at day 75. In the hippocampus, the changes in gene expression were huge early on and decreased around day 21, followed by an increase towards adulthood at day 75. Results from this study demonstrate that there are individual differences in susceptibility to neonatal early life exposure to SSRI paroxetine, with the rats that were genetically predisposed to a high anxiety depression-like phenotype being more vulnerable to the perinatal paroxetine-induced behavioral effects than the risk-taking, aggressive, healthy BHR rats. The genome-wide expression profiling revealed strong region-specific gene expression differences in the developing brain of the BLR rats following perinatal paroxetine exposure. These differences could cause drug-elicited alterations of mitochondrial function, synaptic vesicle transport release issues, and issues with neurogenesis. Prior rodent studies have showed varied results regarding response to perinatal SSRI exposure. Some show that the offspring are not affected, and some show that the offspring are affected. The results of this study can easily explain this. In the previous studies, the predispositions of the rats to depression-like behavior were not taken into account. This study has taken this into account and has shown that rats that are predisposed to that behavior will be affected by SSRI exposure much more than the healthy rats. Adult BLR rats had less serotonin receptors and less serotonin transporters than the BHR rats as well. It is suggested that these differences can shape aspects of behavioral phenotypes in the rats. While we did see a large amount of gene expression change in the hippocampus and amygdala, little is really known about what these changes do or how they actually modify the behavior of the rodents. Innervation density of serotonin could be the major cause of these expression changes. Also, the hippocampus seems to be much more sensitive to perinatal exposure than the amygdala. Findings suggest that dysregulating synaptic vesicle transport and release contributes to a depressive-like phenotype, and that adult SSRI treatment can normalize expression of the synaptic proteins that are involved with transport and release. Perinatal SSRI induced mitochondrial dysfunction in the early postnatal hippocampus when substantial energy is required for cellular remodeling, synaptogenesis, and neurogenesis may represent an important factor that could disrupt normal hippocampal circuit formation, its downstream function, and its contribution to regulating emotional behavior. Lastly, several neurogenesis cluster genes, encode proteins that mediate axonal outgrowth and guidance, potentially indicating that perinatal paroxetine exposure impairs integration of hippocampal neurons into developing circuits. Issues with this study include that the offspring that were already exposed to the SSRIs were drinking the water that was infused with SSRIs after they were born. This means that the subjects were taking more paroxetine than what they were exposed to perinatally, which could throw off the results of the study. Also, the only offspring that were tested were male, even though females are much more likely to be depressed. They did state that this was due to limitations in the gene expression profiling, but further studies that include female offspring could bring about valuable discoveries. Future studies about the gene expression changes could also help us understand how early SSRI exposure can cause abnormalities in offspring. It is uncertain how these results will translate to humans because there are large differences between human behavior and rat behavior.